which is an SFI funded PhD training program. So the CRT, it's a cohort-based PhD program. It has over 150 PIs involved across the 7 universities in Ireland, with the aim of training 100 PhD students over a 7-year period. So in the first 3 months of the PhD's training, we run what's called the residential training program, where all the students attend the University of Galway and undertake training in the fundamentals and applications of genomics data science in human health and basic research, before then going on to enroll in their university of choice to complete their PhD. This webinar serves as a keynote address in part of a week-long set of masterclasses where trainers associated with the African Society of Human Genetics will be delivering in-person training to our cohort of students in the residential training program. So today we have another really exciting talk from Christian Happi, who is a professor of molecular biology in the genomics department of biological sciences and the director of the African Center of Excellence for Genomics of Infectious Diseases, both at Redeemers University. He is known for leading a team of scientists that use genomic sequencing to identify a single point of infection from an animal reservoir to a human in the Ebola outbreak in West Africa. His research focuses on the infectious diseases, including malaria, Lassa fever, Ebola virus, HIV, and SARS-CoV-2. He also played a significant role in the genomics effort, such as the 1000 Genomes Project and H3 Africa. More recently, he has led efforts for SARS-CoV-2 in Nigeria. In 2020, he gave a TED talk called A Virus Detection Network to Stop the Next Pandemic. Prof. Happi is an advocate of open science and is a sitting advisor of Open Therapeutics and its open science platform, Therapoid. So just some housekeeping before we begin. If you could please just keep your microphones muted, and if you have any questions, just type them into the chat. So, Prof. Happi, if you're ready to begin, then we can start.
Thank you. Again, I hope, thanks for enabling me or giving me access to open my video and my microphone. So I think now I can get started by sharing my screen. So quickly, probably just get my screen shared. Just hope, can you confirm you see my screen?
Yes.
All right, thank you. So it's a pleasure, and then I want to thank the organizer for the invitation, and it's a pleasure being here today to share with you a bit of what we do in the space of genomics for pathogen genomics, and then also for human genomics, and then try to understand mechanisms of disease spillovers, and also look at disease, what I call human genetic susceptibility to infectious diseases. Today, I'll be focusing a bit more about how we've been able to use genomics to characterize and then to perform surveillance on microbial threat across West Africa. And from the African Center of Excellence for Genomics of Infectious Diseases. So this center is a WHO, Africa, CDC, and then World Bank reference center for infectious disease genomics research and innovation in Africa. And then I'm the director for, and then I'm also leading the Sentinel program, which is what I gave a TED talk on, and then the moderator actually introduced later on. So that's basically the outline that I would give you. I'll be for my talk. So, and then my talk is going to be more or less about genomic epidemiology and then translational pathogen genomics, more or less. So I think that's basically what we'll be talking about this morning. So as you understand, I mean, as you know very well, genome sequencing has really transformed the way, we we approach healthcare and then research. And that has really brought a lot of benefit in terms of, advancement of medicine and also a lot of economic benefit, and then these benefits, both in health and then in economy has really been benefiting only countries in the North. And then Africa has really been not contributing much to it, or Africa has been really left behind when it comes to, using the tools of genomics. But then we're trying to, but we basically just have realized that the only reason Africa really not contributing significantly to the field of genomics or taking advantage of the economic and technological benefit of genomics is simply because there's no infrastructure, and then there's also knowledge, on the continent. So, and we decided we made this observation a few years back, and then decided to set up a platform really to reduce the gap, that is existing between the rest of the world and Africa, and then try to also use genomics as an equalizer so that we can accelerate Africa development and contribution to this, to the global initiatives. And because Africa really is a country that is so rich, both in natural resources and then also has a very powerful biodiversity, we decided to focus on one of the major problems that plague Africa, and then that has to do with infectious disease. If you look at this map as it's coming up, you're going to see here that, these are, pathogens. These are pathogens that do have pandemic potentials. And if you look at this pathogen with pandemic potential, you realize that, the majority of these pathogens are right here, in Africa. And in real fact, Africa really harbors about 70% of the pathogens that contribute to pandemic potentials in the world. And in the past 30 years globally, there have been 30 new viruses discovered across the globe. And that has also increased significantly in terms of number, that has increased significantly the number of epidemics and outbreaks that we see in the world. But in the past 10 years in Africa alone, the number of disease outbreaks in Africa has increased significantly by 63%. And we have more outbreaks in Africa than everywhere else in the world. So in order to address this problem, we basically set up, a platform called the African Center of Excellence for Genomic Infectious Disease, with initial funding from the World Bank and then the US NIH. And this was really to address local problems and then leveraging genomics technology to start addressing these problems. And then this platform is basically is a network of collaborators and partners, long-standing partners, in West Africa. And then this partnership is built around clinical testing centers and academic institutions in West Africa, but with also international partners, which naturally are Harvard, the Broad Institute, Tulane University, and then also partners in the private sectors like Illumina and then Dimagi and Corgenix to start up with. And really leveraging this partner, we actually set up two broad missions. One was educational, and then the second one was research. So the educational goal of these broad missions were basically to create, a vibrant academic and research environment that transcends national boundaries, where young Africans could come and then do high quality translational genomic research for health development on the continent. But specifically, we wanted to use this platform as a way to build a critical mass of well-trained African young scientists that could leverage the knowledge and the skills of genomics, to a prevention, and or control of infectious diseases. But in order to do that, as I've mentioned before, because we knew there was no knowledge on the continent, so we really created new curricula, first of all, of genomics and bioinformatics that is geared toward, infectious diseases, but also non-communicable diseases. And in addition to that, we engage community regularly in prevention effort and public health education. This educational goal is mainly supported by the World Bank, but all of the goals that I mentioned here, be it overarching goal or you're talking about either the overarching goal or the research component, are supported by the World Bank funding. But then we also, my laptop, for some reason, okay, I'm back here. So, and then the research component is basically supported by the US NIH through the H3 Africa program, and many other funders. So really, we thought about looking into what are those things that really cut across Africa and then where we could use genomics to address. And we identified that fever is actually one of those things that, that is a common denominator to a myriad of diseases. Many diseases present not only in Africa, but globally, with fever. But then behind the fever, then there could be many possibilities. Generally in Africa, when somebody presents with fever, the first culprit is malaria. And if it is not malaria, then we start talking about typhoid, or we start looking into potential viral diseases. So we thought of using this platform as a way to use microbial metagenomics, not only to identify identify characterized pathogen that we know, but also to do pathogen discovery. And in that way, we should be able to come up with what we call a microbial landscape. By having that microbial landscape or establishing a microbial landscape, we could easily develop countermeasures, and these countermeasures could be diagnosis, vaccines, and then therapeutics. And ultimately, by bringing the research component and then the research and then the education component that I mentioned earlier on, we basically will be addressing Africa's critical need for genomic research, and that critical needs really has to do with, as you see this Venn diagram, is actually the genomic capacity building. And that genomic capacity building is really centered around, setting up genomic teaching and sequence lab facilities, diagnostic core facilities, and then also build a sustainable science career for young African scientists so that we can all harness the tools and then the technologies of genomics toward addressing public health or health challenges on the continent. But ultimately, our goal in this program is basically, to build what I call the next generation of African pathogen hunters in the bushes of Africa, and empower them so that they can basically start characterizing the pathogens before they get back to us. You will agree with me that with this approach, we definitely leverage knowledge and skills and technology to start playing what I call offense instead of playing defense, and giving Africa its prominent place that it deserves in the communities of, among other peoples, in the world, because we have a lot to give because of the biodiversity that is also already existing on the continent. And actually, one of the, going downward, I'm going to just illustrate and give you a few cases, how we've been able to use this technology to really not only leapfrog this technology, not only to address continental problems, but also to actually just put Africa in the driver's seat when it comes to addressing public health challenges or use this for public health. The first demonstration of this was actually when we transformed outbreak response during the Ebola epidemic in West Africa in 2014-2016. So during that Ebola 2014-2016, when the epidemic was really biting hard in Guinea, early on, and then the world was where we had a premonition that this could spread and be a big problem for the African continent. So I brought together in my lab at the time in April 2014, a few colleagues of mine, at the Broad Institute, from Harvard, from Tulane, other institutions, and then to start brainstorming on what we could do if Ebola spread out of Guinea and became a very big public health challenge, on the continent. And we identified diagnosis as one of those key issues, because if you cannot diagnose a disease, you can't even control it. So, and what we did was actually to go on and then start training people in Sierra Leone, where we had a very strong footing, and in Nigeria, Senegal, and other countries, to respond to, to diagnose or to detect, Ebola, using molecular diagnostic techniques. And as we rightly predicted, a month later, after we had our meetings, had the first suspected case of Ebola in Sierra Leone, and our team, on the ground was able to make that detection, very quickly, and activate our contact tracing mechanisms. So what really is important here is the fact that within the first week of the outbreak in Sierra Leone, we're able to, we have 35 suspected case, confirmed 14, but then actually link that really index case to a traditional healer who was basically responsible, who claimed that he could manage a patient and was really at the source of, spreading the infection across people. He died along the line, but before dying, he has created a lot of havoc. But what is important and what is interesting about what we did at the time is the fact that, in the first 3 weeks of the outbreak, our group generated the first 99 full genome of Ebola sequence in the world. We not only but we also made the sequences fully available to the international community for use. So, and basically, they also use that as a way to identify malaria and then other diseases that were not Ebola. And this was the very first time during an epidemic when you could use genomics as a way to really start helping not only public health response, but also supporting evidence that, we had Ebola spreading, within the country. And this is very interesting because at the time when we were doing this, organizations like the WHO that was founded in 1945, were still functioning, with the reflexes of 1945, thinking about, how they could get the sample, go analyze after the outbreaks it contained, and then give us an idea of what exactly was responsible for the outbreak way or years after the outbreak was somehow contained. And this is how they've been operating. But we really changed the way, this was doing, the way investigative, epidemiology was done at the time. And it was the very first time, genomic epidemiology was applied in the context of Africa. And then we basically just, demonstrated that this could be very useful. Not only we sequence genome in real time, we use the data to help public health response, but we also made sure that we share data and then in a database. And what is interesting about this story is that all of the things that we put in place, be it real-time genome sequencing, open data sharing, and then also identifying, variants of concerns or what I call a mutation associated with increased transmissibility, and all of the things that we set up in 2014 is what the world used today in order to address the COVID pandemic. So when you hear about GISED, you hear about variants of concern, you hear about, doing genome sequencing in real time. We pioneered that in 2014. So that really give us a lot of pride to know that the project, the one thing that the tools and the technology and the approach we established in 2014 has become universal. And we need to be very proud of the fact that, we led this whole initiative. So, and what is, going forward, then you will see that we did this very quickly. Imagine in 2014, so when genome sequencing was in its infancy, at the time, we were able to actually create, generate data from sample collection, to submitting to GenBank in 10 days. That was really super quickly. We were not really interested in publishing. I can tell you that we generated hundreds of genome sequences and did not really publish our data, until probably 9 months later. So really that gives you an idea of how we really thought about equity in this case. We're really not looking for being famous, but also thinking about how we could leverage, information to respond to the public health first before thinking about publishing. So it wasn't an academic exercise. It wasn't, as people may think, but it was really more how you could use data to respond to public health and how an academic group could actually use information to help public health response. We established for the first time, in the case of Sierra Leone, for instance, how you could have different variants entering the community, different lineages of Ebola virus in Sierra Leone. 4 different lineages entered the country, and then you could see tracking over time. You could see how by June 16, we have this date where one of the variant or one of the lineages, SL3, took over. And this became the variant that actually started transmitting more and then overshadowed every other variant that were actually present in the region at the time. But then the question was, what was responsible for increased transmissibility of this variant, and why did this variant take over? And then you could see here, this is the first demonstration again during an outbreak when we actually started looking into that variant. We identify quite a number of mutations, and then we identify, about 21 mutations that were fixated around the very core of the virus. And this was increased, with what we call in vitro analysis and then functional analysis. We demonstrated for the first time that this variant, the mutation that accumulated around, the receptor, around the surface protein of the virus was associated with increased transmissibility. That was the very first time this demonstration was made. And we're glad that, that same approach that we used in 2014 is what the international community or the global health community used this time along to characterize the different variants for COVID-19, and that has become the global standard. So, but then we also did something that was unprecedented. As you look on this map, basically just looking at genomic sequence data and then establishing transmission chain, and then also showing clearly how viruses were transmitting from persons to persons, from country to countries, and then from town to town, district to districts, and then also from provinces, and then actually from countries to countries. And this was again showing, and this really reveals some very powerful, provide powerful information, and then actually gave us some viral genome, revealed some very powerful factors that were actually involved in spreading and sustaining the transmission in the region at the time. We didn't just stop there. We made some tremendous observation during the outbreak. We realized that, people who had simple malaria, who could go to Ebola triage center because they have a fever, but they also got infected there and then took Ebola back home and then start spreading it within their family. Or we realized that the diagnostic time was too long. It was taking, days and days, to respond to, to provide, to give diagnostics to people that had mere fever or people that have Ebola fever. And then we really went on to translate, our genome sequence data that I mentioned to you into the point of care diagnostics. And we were the first group to actually develop the first Ebola rapid diagnostic test, that received approval for emergency use authorization by WHO and the US FDA. And then the introduction of this test actually proved to be very crucial by flattening the curve or bending the curve, as we would say, for the outbreak, because within 10 minutes, doctors or lab scientists could know exactly whether a patient that had fever had Ebola or the patient had none, or a patient that had Ebola had cleared and then could actually be discharged. But then this came to bear tremendously when it came to Nigeria. You could see here, my lab in Nigeria at the time was located quite about 2 minutes from where you have this hall. This is a crowd on a Friday in Nigeria, where at the Redeemed Christian Church of God, with our publicity here, showing a crowd, you have more than 3 million people gathered, in a place and then a place of worship. So the question that came to our mind at the time is that when you have such a gathering, what will happen if few people within this crowd had Ebola? And we're very worried and very conscious. So we actually got prepared, did a lot of sensitization. But as we rightly predicted, we're also very ready in Nigeria. Then, on July 21st or July 19 to be precise, I had a call at late in the night where we had a suspected case of Ebola in Nigeria. Then the sample was sent to a lab on the 20th, and then we really did an analysis very quickly and then gave the result. And this really was the first case of Ebola in Nigeria, diagnosed within 6 hours. And then we shared the data with public health authorities and started coordinating with them in order to contain the outbreak. But then it shows you the power behind the technology that we have, because, working with public health authority, we're doing genomic sequencing. You have contact tracing here, you have genomic sequence data here, you could superimpose these two curves. But what is important is the fact that, with speed and accuracy, in the case of Nigeria, we're able to contain this outbreak in Nigeria, a country of 230 million people, and an outbreak that started in Lagos, a city of more than 25 million people. We contained that outbreak in 93 days with only 20 cases and 12 deaths and 8 deaths. That really is unprecedented. Actually show the power behind this technology. So the next use case I'll talk about is actually a disease called Lassa fever, which is a viral hemorrhagic fever that kills a lot of death, was first identified in Nigeria in 1969, and it is responsible for a few thousand deaths of people in West Africa every year. But what is important about this is the fact that, for almost 40 years when this disease was basically diagnosed, there was no diagnosis in Nigeria. And we did the very first molecular diagnosis in Nigeria about 37 years after the virus was diagnosed. Before we actually set up the first molecular diagnosis, what used to happen is that people that got sick used to be, a sample used to be sent to Germany, and it took about a month, if not more, to diagnose. And 90% of cases, a patient at the time, died before the result of the diagnosis came back. But then, just using, setting up the molecular diagnosis and then also doing genome sequencing actually proved to be very powerful in containing this disease, or in trying to actually eliminate this disease as I speak. So we went on and then, I set up this molecular diagnosis and then actually did the first diagnosis on site, in Nigeria for the first time. I just want to point here that when I got to Nigeria in 2007, around the time, there was a hospital, and the name of the hospital is here, and then a bush. There was no diagnostic facility in the hospital. We set up this small hut over that you see here, and then a few months later, we brought up the diagnosis. We even handed over to the hospital, but trained the hospital facilities in order to do the diagnosis at the time. And then we did the very first diagnosis on site on September 27. And then from there, also helped to set up a clinical ward for managing patients. But I can tell you today that this is a lab, from this little hut that you see here. This is a lab that we built in this place. I mean, a modern facility, but we've also used a platform as a way to train, people in that hospital. And then that hospital has gone from being a small a hospital that nobody knew, to become a center of excellence for clinical management, and then also for diagnosis of Lassa fever, and then other viral hemorrhagic fevers, in Nigeria. But then we went on to do the very first full genome sequences of Lassa fever in the world, creating the largest catalog of Lassa fever genomes. But then in addition to that, we're able to, I mean, that was the first time we used unbiased genome sequencing to do this. But in addition to that, that revealed us very important things. One was the fact that, we identified that that the Lassa fever virus, emerged from present-day Nigeria about 1,185 years ago, was circulating in Nigeria about 600 years, and it moved out of Nigeria about 400 years ago, toward West Africa. But then this really were very important because, apart from just unraveling the ancient origin of this virus, then we really also understood that there are several lineages circulating depending on where you are in Nigeria. In the north, were lineage 3, and then the first lineage also emerged from Lassa. Then in the south, you had lineage 2. But in addition to that, we translated all of the genome sequence data into point-of-care diagnostics, and then we created what we call a pan-Lassa, rapid diagnostic test that can identify all the different variants or the clades of viruses that are circulating down. And then you could see that here. So again, showing the power of translational genomics, and this has been very helpful in terms of helping, a diagnosis and then also advancing science. I want to state here clearly that between 2015, when we actually finished the diagnosis, and now, these are accelerated vaccine development. Our discoveries have accelerated vaccine development, but I also help a lot in the sense that now there are 2 vaccines undergoing clinical trial in West Africa. So again, this is another, use case whereby in 2018, Nigeria, within the first 8 weeks of the year, experienced a 200-fold increase in the number of Lassa fever cases and number of Lassa fever deaths. There are a lot of unanswered questions. How could you in 8 weeks have more Lassa fever cases and deaths than you have in the past 2 years combined? There are a lot of unanswered public health questions. For instance, were we dealing with a new variant, or was it Ebola that was back, or we're dealing with human-to-human transmission, or that the diagnosis were not effective? So what we did at the time that we actually used real-time, genomic sequencing to respond to the problem. Collecting samples across the hotspot, we were able to sequence and actually work with the Nigeria Center for Disease Control, and we're able to establish one thing, and then doing real-time genomic sequencing in real time, we're able to demonstrate one thing, is that, it wasn't due to a new variant or to something else. So we actually show no evidence of particular new viral strain. We have no evidence of sustained increase and sustained human-to-human transmission. We identify some clusters within hospital. These clusters were within 2 to 8 patients, and then this through nosocomial transmission. Yes, we acknowledge that, and this really was used to actually help the hospital to actually be more careful and reduce nosocomial transmission, but then also that helped to save total healthcare patients. But then we really saw that the search was actually associated with the introduction of multiple introduction of genetically independent viruses, similar to the lineages that we know. But one important thing is that we identify, what we found out is that, lineage 2 that I mentioned before are split into 2. We have 2A in the west and 2B in the east, and then lineage 3 were in the north. So invariably, Nigeria was dealing with a crisis whereby Lassa fever in the north was a bit different from the one in the east and in the west, but then, because they were driven by 2 different lineages. And this really led us to start understanding, to say, oh, well, if this is really being transmitted from rodent, to human, this finding we made it in the New England Journal of Medicine. If we're able to do this, it is clear that because rodents were transmitting this, then we really started to start looking at genetic diversity of the virus in rodent and see how that relates to human. And we got our teams in the field. You see them trapping the mouse and then sequencing in the field here. But then we're able to establish for the first time in the history of science how you could take a virus sequence in the rodent and link it up, with an infection, in the same house. So, and these were cases where we identified the same virus in human, and then full genome, and then identified the full genome, the same virus in a household, where a rat was cut. So identifying the same virus in a rat, and then in a human in the house where the rat was cut. And that really was very helpful to really establish that. But we also leverage, advancement in genomics and technology in order to move away from the rapid diagnostic of antigen capture test, as you know very well, that has issues with sensitivity, to actually develop a CRISPR-based diagnostics, that is applied and use gene editing to identify guide RNAs and then and create a novel diagnostic, rapid diagnostic test for Lassa fever, and then basically using, Cas13 instead of Cas9, as you know. So, and this really became revolutionary because at least with this, we're able to not only diagnose Lassa fever in real time, but also, faster. But actually, this was the very first, CRISPR application was CRISPR to an infectious disease pathogen. So whereby, use machine learning, we identify the guy's RNAs, and then we create the, sequences that are very unique to the variant or to the virus, create a guide RNA, and then actually use tag endonucleases with reporters, as you see here. And then once you have a cut, then you have a diagnostic like a plus or minus, as you see here. This is just the conventional thing. But then the difference here is more or less like, you are doing and doing this here, help in the sense that it helps in not only just doing the diagnosis quickly, but actually make the diagnosis a bit faster. And that's what you need, in the context of an epidemic or the concept of an outbreak. So, and then we got Amy Maxson from Nature, who came and embedded in our lab, and I was in our lab for 5 days to see this whole process, and then this had published, we had this published in Nature, and this was the very first time, that you could do. This is relatively cheap. You're talking about a $3 diagnostic compared to a $130 PCR test. So, but then we also had a lot of, we leveraged our work to start working, to also work on the human genome side of the whole story. And then we basically just have we were involved in the 1000 Genome Project. We work in the Mendes of Sierra Leone, the Ishans of Nigeria, and then the Yorubas of Nigeria. What is interesting about this global project, where we're talking about, creating the reference for the human genetic variation, is that the population on which we work, which are the Mendes, and then the Ishans of Nigeria, and then the Yorubas of Nigeria seem to be the ones with the most genetic variations across the globe. And that really started making us believe about, thinking about how infectious diseases actually could drive, the diversity that we see, or the genetic variation that we see in African population. And that really made us to start thinking very widely, to start thinking about what are those signals that could exist in African genomes, in relation to a disease like Lassa fever. So we performed the first genome-wide scan analysis of people, with Lassa fever, and then those living in the same environment without Lassa fever. And then we found something that was very extraordinary. The genome-wide scan actually enabled us to do a whole lot of things, in the sense that this was built on observation, that we go into the same community. Some people get exposed to Lassa fever and others do not get exposed. And then you realize that some that get exposed do not get sick, while others that get exposed get moderately sick and others sick. That really enabled us to do the first GWAS study, for Lassa fever. And this is a 10-year study. So, and we were able to identify on chromosome 13, on this locus that you see here, identify, on the large gene, for instance, some mutations that we now are very certain, and then this paper is coming up in Nature soon, so associated with resistance to Lassa fever. So, and then, our hypothesis, is really the fact that, a signal that large may indicate an evolution of resistance to the Lassa fever virus, and this is what the data actually prove us. But then we took advantage, of a previous finding for Stephen Cohn's, and into the piece to his memory, who left us, about a few years ago. So where he basically just looking at, post-translational modification of acetylglycan, which is a cellular receptor that interact with adenovirus. And we absolutely observed that Lassa fever being an adenovirus, and then that really led us to start looking at this receptor very critically, and then really start looking at what could this receptor do. And what we really observe in our functional analysis and in vitro studies, and then is the fact that, alpha-dystroglycan is this receptor that is actually involved in the entry of the Lassa fever virus, into the cell. So, but then alpha-dystroglycan, to be effective, actually have to couple with large protein, and then the alpha-dystroglycan large, complex is really what is very efficient in actually bringing the Lassa fever virus into the cell. But when you have a mutation at large, the way we've seen in these people that will have these mutations, even though alpha-dystroglycan coupled with large, then that blocks the mutation at large, blocks the entry of the cell, of the virus into the cell. And this is our very recent finding in our lab. And then that's what I'm saying, that this really has demonstrated, the power of this technology coupling to human genomics, to understand exactly how this So this has tremendous implication in the sense that we now have, we definitely have the potential now to actually take it forward by creating either molecules, that will block, the virus from getting into the cell, or that will actually prevent us from getting infected. So the next thing I will talk about is actually to see how we've been able to, use clinical observation to make the largest metagenomic study in Africa. So working in areas like ERA where we work, we used to work with clinicians, and then the clinician would tell us that, well, you have all the molecular diagnostics, but unfortunately we see patients that present with diseases like similar to Lassa fever, the molecular tests are negative, but then at the end of the day, when we treat them with some of these antiretroviral, then it seems to work. So it's either that you have other viruses that are presenting like the ones that you are working on, or there are other pathogens that are also presenting clinical symptoms like that. And that really made us to start, in the early years, 2010, to think about how, 2011, to start applying that technology on metagenomics. And then we really conducted the very first the largest metagenomic study in Africa. So, and then we did a case control study where we got people that are healthy in court, and those with febrile illness, try to identify what are those drivers. So, and we're able to identify in that process, identify novel rhabdoviruses, discover novel rhabdoviruses, 2 novel rhabdoviruses in people. So, but what was really intriguing for us was the fact that instead of identifying them in this group, you could see in this group, you see so many people that have more viruses than the one in that are healthy, but we identified them in people that are supposed to be healthy. We went back to these folks and they were trying to find out, what could be probably a little bit more. And we realized at the time when we collected the sample, they actually were ill, but then they took antipyretics, and then that suppressed the fever. They appear to be normal, but then in reality, they're having the virus. So we use, we named those viruses Ecomavirus 1, AKV1, and Ecomavirus 2 to follow the global nomenclature, at the time. But then what is important here is the fact that when we use the sequence data to create, an analyzer to actually look at the exposure of this virus, we observed that, about 80% of the people in the communities where these 2 viruses were discovered had exposure. So this really buttressing our point of the fact that, viruses that are circulating are much more older than what we imagine. And then we should rather be talking about emerging diagnostics instead of emerging diseases, because those viruses are ancient. But what is interesting about these 2 viruses is that they share very strong similarity with the Bakongo virus that was described in the DRC in the year 2012, and that was responsible for an outbreak of viral hemorrhagic fever there. So, and this is another use case. Here we use real-time metagenomics to actually save the situation in Nigeria one more time. So in this case, for instance, in 1 week, Nigeria lost 179 school children dead for a disease, for a mysterious in-court disease. All molecular and then ELISA testing proved to be negative. And then the Nigeria Center for Disease Control referred about 22 samples to a lab because they didn't know exactly what was the reason. So people were panicking. So within 72 hours, we established that it was a yellow fever, yellow fever. But then the yellow fever that you established here, you could see them clustering, all of them here, were different from the previous yellow fever strain described in Nigeria that you see here prior to in the 80s and in the 70s and in the 90s. So, and that for us was very, very intriguing. But what we noticed is that this particular cluster here was different from the ones that have been circulating in Nigeria in the past 94 years. So this information was shared immediately with the Nigeria Center for Disease Control. They also immediately declared an outbreak. And then together with the National Primary Healthcare Development Authority, they were able to contain that outbreak within 2 weeks, saving thousands of lives. Again, showing here how we use real-time metagenomics or real-time genomics in order to save, to impact, public health and then to transform the way it happened. This is another use case, for instance, in Nigeria, whereby in a city like Lagos, as I mentioned, 25 million people, one of the largest hospitals called General Hospital Ikorodu, for instance, parents present with a child that is about 2 years, bleeding from all orifices, from the nose, from the ear, from the mouth, everywhere. In the pediatric ward. The pediatric ward, gets empty within minutes. Everybody take their patients away because they believe Ebola is back. The pediatrician all disappeared, and because everybody is camping out for his life. So, and we sent our team down there, collected the samples, and in 72 hours, we established that it wasn't due to Ebola, but it was rather due to an enterovirus B, with heavy viremia in the child. That provided confidence in this hospital, and then they were able to give me back, and then continue, managing patients the way they've been doing. The next use case is, again, has to do with another, I mean, with the bacteria. You know, we had cases whereby you have treatment of patients with 2 patients, you know, with different kind of antibiotics, and then the treatment appeared to be negative. I mean, diagnostics by molecular diagnostics in the lab actually show that it's actually MDR-TB, you know, but then by the time gene experts show that is MDR-TB, but by the time the samples are sent to a lab, we actually found that this is not MDR-TB, is actually extensively drug-resistant tuberculosis. And this really, here we reported then at the time the first 2 extensive drug-resistant tuberculosis, not only in Nigeria, but in West Africa. And this information were very useful because along the line, one of the patients died along the line, but actually this information revealed the fact that we have extensive drug-resistant tuberculosis in Nigeria, and then we should be very careful. So what did we do with all of the information that we gathered? I just want to say here that we're doing this in the space of not only human, we also have, we are using a walnut approach, you know, to address this problem. So we're doing a whole lot of work in animals, we're doing a whole lot of work in human, but then what we've done is that we've actually used all of the information that we've had to create, even before COVID-19, a pandemic preemption and response system called Sentinel. And this is between myself and my colleague participating at the Broad Institute, but then it's really built on 3 pillars. One is actually early detection. You agree with me that if we have the ability to detect pathogen early, then we can actually contain epidemics. So, and it's early detection that is creating more genomic tools and novel next generation diagnostics, like the one we've created, CRISPR-based diagnostics, you know, to detect pathogen very early. And then that is in the remote areas of the world within hours. If that test proved negative, we send them to, we've also created novel tools, you know, in hospitals. And here we call this the MCOMN. So whereby you combine about 465 different pathogens, where you could actually detect the nucleic acid in one single test, and you could multiplex this in 86, 96, 384, and then 1,500 and something if you want. So, and you know, that enables you to really have a one-stop shop where we could detect a myriad of pathogens in one single stop. And then all of this information at each point, you know, are transmitted in real time to public health, you know, I mean, to public dashboard, so that with GPS and all kinds of clinical data and metadata, so that they can know exactly what to do when they have an outbreak. So we also have a set of genome centers, you know, whereby samples are sent to the genome centers, and then we could do real-time genomic sequencing and then share the data with public health authorities so that they can actually use information, you know, to address public health response. All of this data, and then, you know, and then information technology tools that we are developing, you know, are handed over to public health frontline healthcare workers, and we're empowering them to use novel genomic diagnostic technologies, and then data sharing tools in order to capture the data and make it available to public dashboards, both locally and at a national level and at an international level. So basically, this describes the Sentinel program that we should build on a hub and spoke approach, and that has 3 major pillars: detect, connect, and empower, that I just described here. So that really enable you using the hub and spoke approach, where you have the genomic hub, they're dealing with the spokes, you know, and then the spoke deal with the most remote places, then to create a system, you know, that is really interconnected, you know, and then that through that connectivity, we can actually respond to things in real time with all of the data. You agree with me that there are 2 things that you really need to address an epidemic. One is speed, second is accuracy. Once you have those 2 things, you could really just respond to the epidemic. But then finally, you use that to empower frontline healthcare workers and public health authorities to respond in real time. And that's what we've really done. Over the years, and then that has come to bear, you know, during the disease epidemic. Ultimately, the goal for us is really to see how you go from high genome sequence facility that we have, creating throughput diagnostic, you know, technologies, you know, using CRISPR-based diagnostic, for instance, work with communities in order to address, you know, to start creating that partnership that you use in order to address public health response. But in doing this, you want to expand the scientific capacity, you know, and enable sustainable independent surveillance outbreak, you know, for epidemics, you know, in Africa. So, and this really what we practice and particularly during the epidemic outbreak. During the epidemic, the COVID-19 outbreak, epidemic in Africa. Remember that, the epidemic of the COVID-19 in Africa started in China and then spread across the world. In Africa, Egypt was the very first country to report, then followed by South Africa. Then Nigeria was the third country to report that, report the virus. But what made the difference between Nigeria and then Egypt and South Africa is the fact that we had all of the technology and the skills and then the know-how on the ground. And what happened is that as we received the sample, within 48 hours, we were able to elucidate the full genome of the virus and then show that the virus was imported from Switzerland, and then it wasn't from community transmission within the country. But then really that sequence that we did at an unprecedented speed of 48 hours became the very first SARS-CoV-2 genome sequence in Africa, and that really gave confidence to African researchers and African public health institutions that this is doable and that we could actually use it to respond to the epidemic. The rest of the story, you all know how South Africa, leveraging what we've done, was able to actually sequence the first beta variant and then Omicron, really giving Africa its prominence, in the communities of nations. And we use such, for instance, to help public health authorities in Nigeria to respond to epidemic. You could see here where we created the dashboard. You could see in real time, the different strain and variants circulating in Nigeria over the time and over the years. Over the year. And this information is really what Nigerian public health authority were using to respond to the epidemic. When you hear, at the time when you used to hear, for instance, the head of the Nigeria Center for Disease Control, Dr. Chikwe, said that, no, we are guided by data. These were the data behind that he was using, because we used to make an update or weekly update, to the Nigeria Center for Disease Control, and they use it in the Presidential Steering Committee every Monday. But, we knew at almost in real time where different variants were circulating in Nigeria. We knew at some point that in Nigeria we had 49 different lineages of variant of SARS-CoV-2 circulating. We knew exactly where these were, identifying hotspot, giving information, and they use it as a way to really guide the response. But then you could see here, for instance, over the years where we were tracking different variants circulating in the country, and then when a variant comes in, when it emerges, when it takes over, and all of that. So, but then what is important is the fact that we also translated all of this information right from day one. I can tell you that within 8 weeks of the outbreak in Nigeria, we went from molecular diagnosis to create this diagnosis, called SHINE, which basically is more or less doing CRISPR on a sheet of paper. We lyophilized all of these things. So, and then what is important about this is that more or less you are doing a PCR on a sheet of paper. It gets very cheap, $5 a piece. But then at the end of the day, it can be done by if my grandmother was living, I'm sure she would have been able to do it, because you need no knowledge. What is important is also the fact that, it works with saliva, nasal swabs, and then, and even the urine version. So these are the very first tests that we did in our lab, but then this really also has been used. As I mentioned before, so now we have the ability to multiplex all of these diagnoses in one single place. And then we've called what we call the MCOMN here, which is a CRISPR-Cas testing, massively multiplex test. And as I said before, this is a version where you could do, 169, but now we are pushed it beyond boundaries, so you could do up to 460 different pathogens in one single test. And then you get the result, within minutes. We've also leveraged this to train and build tremendous capacity across Africa. So you could see here, for instance, this is our team across, training people in different labs in Africa. So we've been able to train over 1,300 people through short-term, and a degree program, partner with Africa CDC, the Nigeria Center for, and the ministers of health across the continent, the WHO, Afro, to train a critical mass of well-trained African scientists. Here you could see here, in a small place, a small lab like ours, we've trained people now in over 42 different African countries. And these trainings have been proved to be very impactful. This is in collaboration with the PGI, the Africa CDC, for instance, and the WHO Afro. It's over 42, and then all of the training that we've done has been able to increase dramatically the number of sequences that were generated from the African continent. You could see here, at the end of 2020, Africa had only about 5,000 sequences. But with all the training that we provided, along with other centers like the Institute Pasteur de Dakar, and then, for instance, also the SERI and then CRIPS in South Africa, that increased the number of genomes in Africa, to over 100,000 within the space of a year when we're doing this. But that also gave the opportunity, for instance, for countries to identify, like South Africa, to identify the beta variant. In Nigeria, we identify the eta variant. which was probably a variant of under interest. So, and that also tracking the movement of all of this variant across, across the country, across the continent. And that really is very revealing. But we created dashboards like this one, where, public health authority, you could just basically look at the tree. You don't have to be, you could see sequencing, click on it, you just look at the tree. You don't have to be a bioinformatician. It gives you the tree, and then you can also look at the map. It gives you the different variant, and this is a case of Nigeria where we're piloting this, whereby you see the different trees. You see Lagos, for instance, you could see the variation over time, the different lineages and variant. You could identify the hotspot in Nigeria at a given time. And this is really what public health people need in order to really move the field forward. So, at this point, I just really want to say that, yes, we did all of this, but we also leverage our platform to also make a call, for equity, as we work in the field of genomics for public health, for instance. And recently, we really scored at the WHO and many other international organizations to be a bit more, less discriminatory and non-stigmatizing, especially when it comes to nomenclature. And this was for monkeypox, because monkeypox, the present monkeypox epidemic in the world is in Europe and not in Africa. But actually, they were trying to bring it down to Africa. And then we made a call together with colleagues over in the UK and then in Brazil and then in others, really to make clear that we need to be less discriminatory and then less stigmatizing when it comes to giving nomenclature. Let's use things that are less neutral and then make sure that we can address, we can be more responsible, so that together we can actually create what I call a global equitable movement, where people can report sequence data and report things to prevent epidemics in the future. So at this point, I really want to really thank my colleagues, thank my collaborators, and then also the funders. Thank you.
Thank you, Prof. Happi. That was fascinating how quickly you can go from a suspected case to a diagnosis. So just while we wait for some questions to come through, I was just wondering, is the protocol for when you identify an outbreak with a potential to become pandemic, is that fixed across different nations, or is it kind of a unified framework that's rolled out to?
Well, I think what we did at the time is a unified framework, because when we started this in 2014, it was something new. Nobody thought about using genome sequences actually to respond to outbreak, but we're very happy that it has become more or less like a unified protocol, and that cuts across the globe, because that was the first time in 2014 that this could ever happen, that we were doing this in real time. And then we're glad that, it has become the standard. And it has become that thing that now when there's an epidemic, people can quickly sequence, people can quickly share data, and then public health, public health and policymakers can actually use the data to respond to outbreak and to epidemics.
Great, thank you. You said the chat is quiet. So if there's no questions, I think we can finish here. So thank you once again, Prof. Happi. That was very insightful.
Thank you.
So we're back tomorrow at the same time, so 1 p.m. Central African time. So I hope to see you all again there.